Assalamualaikum girls. Today we have discussed exercise even, and we have discussed this corona case theory and law principle, and we also I'm repeating that we have discussed the whole exercise, but one of the few topics left were data organization, data analysis, and mathematics as an integral part of scientific process. Don't get disturbed from these pictures. They are just for your journal knowledge. Inform you something. This flow chart, I haven't taught this, although it's just for like a flow chart mentioned in our book, that first of all, you have to make observations, what you are doing and seeing in the nature, from your own experiences, from your own thoughts and reading, and then you think about different questions, different problems what we are facing in our society. Uh, then you try to formulate the possible answer to that problem, and that formulate a hypothesis which will be a general solution of the problem. Then you want to make testable predictions or deductions. If my hypothesis is correct, then I expect this, and this. Then we go to gather data to test predictions. How can we collect data? We can collect data by performing experimentations, by doing formal experiments through testing to verify our results. And after doing experiments, we go on to refine or alter, expand or reject hypotheses. And we can go on to develop general theories. General theories must be consistent with most or all available data and with other current theories. So this is a general pathway of how to solve a scientific problem. So now I'll move on to the step of data organization. अदन बेटे आप उधर लेट जाओ, लेट जाओ शेज़ाद, हाँ लेट जाओ ऐचे सही. अच्छा जैसे मैं क्लास लेने लगती हूँ तो पता नहीं मेरे बच्चे कैसे जाग जाते हैं. So data organization. Now data organization is a very important step in a scientific process. Scientists collect and organize data to test a hypothesis by experimentation. So when are we actually collecting the data? We are collecting data during experimental procedure. Otherwise, how can we collect data during hypothesis formulation? So we have to find out certain ways to collect data. Before performing any experiment, scientists describe data collection methods. I mean, जब हम observation कर चुके होते हैं and hypothesis formulate कर चुके होते हैं, and we are proceeding to do certain experiment, then we scientifically describe our data collection methods or our experimental strategy, that what kind of experiment I will collect my data in this way, and I'll make something, whatever you are going to make in this, following this procedure, and then I will test those things by test number 1, test number 2, test number 3. So these tests are the one where you are collecting data. Actually, somehow I'm relating it with my research work, which was on, as it's not a regular class, otherwise I would have described it in detail. It was on hydrogel beads in a different composition. आपने वो Chinese beads देखी होंगी, when you immerse them in water they expand like a balloon. वो तो बहुत ज़्यादा होता है ना, artificial है चूँकि. But in natural biological material हो, जिसे कहते हैं biodegradable material, I mean living material हो तो वो degrade भी हो जाता है naturally. तो चूँकि आपको ये terms पता नहीं है, so that's why I'm trying to avoid them using. अच्छा, तो बायो फ्रेंडली मटेरियल से आपने एक बीड बनाई, एंड व्हेन यू इमर्स इट इन वाटर और certain human solutions, it's going to swell. So those beads can be used to deliver drugs, can be used to deliver certain kind of antibiotics, right? So I was working on that. So I have informed the method to make these beads, and then I have mentioned that I will do this test, hydrogen swelling test, and अब मैं सारा भूल गई हूँ. X-ray crystallography, SEM analysis, scanning electron microscope analysis, which you don't know yet. The electron microscope analysis, और उसके अलावा there were many other tests. So you have to mention all those data collection methods to ensure the quality of experiment. See, the more the experiment is better, the more you're going to mention all these methods. These are the best ways to do any scientific procedure. So data is then, when you collect the data, you organize it into different formats like graphs, charts, again charts and tables and flow charts and pie charts. I'm just describing journal charts. इसका टाइटल देख लें, इसके table, flow chart, pie chart, any kind of maps and diagrams. You express your data in a simplistic way. क्योंकि अगर आप, फॉर एग्जांपल मैंने टीबी के पेशेंट्स पे काम किया था, it was a work on 200 patients. तो you अगर आप 200 पेशेंट्स को अलग अलग डिस्क्राइब करना शुरू कर दें, वन पेशेंट नंबर 1, then patient number 2, then patient number 3, 4, 5, 6, how many a person can read? I mean, it becomes boring. So you have to summarize your data into different charts and tables and flow charts and pie charts. This is what data organization is. You have to summarize your data in a meaningful way. जैसे हमने मॉलिक्यूलर लेवल ऑफ ऑर्गेनाइजेशन में पाई चार्ट पढ़ा है ना, कि इतने परसेंट ऑक्सीजन है हमारी बॉडी में, इतने परसेंट हाइड्रोजन है, इतने परसेंट कार्बन है, इतने परसेंट नाइट्रोजन है और अदर एलिमेंट्स हैं, राइट? रिमेंबर, it was easy to understand. It was the way which makes us easy to understand, right? So this is what data organization is. Let's move on to the next topic, is data analysis. As the name shows, you have to analyze data in different ways. But the thing is that, how can you analyze your data? Obviously, by using certain mathematical or statistical techniques. It is necessary to prove or disprove a hypothesis by experimentation. It is done through the application of statistical methods. You can apply different statistical formulas and techniques during experimentation. For example, the easiest way of mentioning the statistical method is to tell you about ratio and proportion, because ratio and proportion ये जो है, ये आप बचपन से पढ़ते हुए आए हो. Ratio and proportion. The early childhood maths includes ratio and proportion. It is because you can easily understand how scientists analyze the data, because you know ratio and proportion. Other statistical methods are not, you're not familiar to them. So what was ratio? It's a relationship between two numbers and is expressed in terms of quotient. A is divided by B, or A colon B is the way it is expressed. Ratio is expressed by putting this sign or this sign. So it is actually the ratio of one number to the other, trying to find out a relationship between two numbers. Let's take an example. The ratio between 50 malarial patients and 150 normal persons is 1 into 3. 50 colon 150 करें, 50 colon 150 करो, तो 5 on the 5 and 5 3 the 15. The simplest ratio we get is 1 into 3. whenever we find out a ratio कि 150 नॉर्मल इंडिविजुअल थे और उनमें से 50 ऐसे थे कि जिनको मलेरिया हुआ, तो अगर हम रेशियो फाइंड आउट करें तो हम कहते हैं वो, अक्सर आपने शायद सुना होगा कि out of 10 individuals there are 2 individuals who are suffering from anemia, or out of 10 individuals in Pakistan there are 4 individuals who are suffering from anemia, or out of 10 there are every 2 persons are suffering from hepatitis. अब मैं ये अपने पास से बना रही हूँ, it's not the real data, I'm just telling you. Like आजकल के केस में अगर अल्लाह ना करे कहें कि out of every 10 individual 2 are suffering from COVID-19, अल्लाह ना करे, so this is the ratio. It doesn't mean कि हर 10 में से 2 को हुआ होगा. जो जनरलाइज डाटा आपने कलेक्ट किया है उसके मुताबिक आप रेशियो फाइंड आउट कर रहे होते हैं. बट the more your data is, the more huge your sample is, the more reliable your results will be. Like अगर आप 100 इंडिविजुअल पे रिसर्च कर रहे हैं तो वो रिलायबल नहीं है. 1000 पे, 2000, 50,000, पूरे पाकिस्तान का अगर आप डाटा कह रहे हैं, अगर 1 मिलियन पे है, 2 मिलियन पे डाटा कलेक्ट हुआ है, तो then the results are more reliable as compared to just 100 individuals. जितना बड़ा सैंपल होता जाएगा उतना ज़्यादा डाटा रिलायबल होता जाएगा. जब आप मिलियन इंडिविजुअल्स में से रेशियो फाइंड आउट करेंगे कि आउट ऑफ 1 मिलियन 50,000 इंडिविजुअल्स सफ़र फ्रॉम दिस डिजीज, जैसे आजकल निकाल ही तो रहे हैं, कह रहे होते हैं कि दे आर ऑलमोस्ट 2 lakh, I don't know what the current rate is, but 1,50,000 people have suffered from COVID-19. बट उसमें से जो लोग रिकवर हो गए उसको माइनस ही नहीं करते, जो लोग डेड हो गए उनको भी माइनस नहीं करते, तो फिर तो नंबर बढ़ता ही जाएगा ना. Whatever the picture they are representing to us, I think so they are not deducting recovered patients from it. Whatever, but these days the number has reached to almost 2 lakh. I don't know. I'm not in touch with the latest data, but you can find out the ratio according to our population. तो उसको अगर आप डिवाइड करो टोटल पॉपुलेशन से तो रेशो बहुत कम होगी. इतना भी उस तरह से डरने की ज़रूरत नहीं होती जब आप उसको पूरी पॉपुलेशन के साथ कंपेयर करो. बट रावलपिंडी का डाटा या इस्लामाबाद का डाटा विद इस्लामाबाद की प्रोपोर्शन इज मोर डेंजरस. रावलपिंडी का डाटा विद टेकिंग आउट द रेशियो विद द पीपल लिविंग इन रावलपिंडी विल बी मोर डेंजरस, क्योंकि पूरे पाकिस्तान में तो बीमारी नहीं फैली ना. समझ आ गई दैट फ्रॉम अप्लाइंग the principle of ratio we can easily simplify our data. And कितना simple है ये maths ratio, but we can simplify our data with just ratio as well. Let's move on to proportion. It means to join two equal ratios by the sign of equality. I mean proportion means the sign of equality or 4 dots, but actually it is serving the purpose of equality. So what is proportion? It means to join two equal ratios. Okay, I mean essay. Let's look at the example of proportion. A colon B, A ratio B, proportion C ratio D. You are equalizing the two different ratios. So you know that proportion symbol can be represented by the sign of equality or 4 dots. Okay? Let's take an example of proportion to understand this principle more. अच्छा, इस क्वेश्चन को ज़रा गौर से देखिएगा. इस तरह के क्वेश्चन हम बचपन में बहुत ज़्यादा सॉल्व करते थे. एंड आई रिमेंबर मुझे बड़े मुश्किल लगते थे. कर तो लेती थी लेकिन यार ये करके एक्स वाला वाज to find out, knowing the 3 values and finding out the 4th unknown value was very easy, but still it was very confusing in childhood. I don't know why, because children's mental capacity is not beyond, I mean काफ़ी कम होती है, शायद इसलिए हमें वो बहुत मुश्किल लगता है. Let's read it. A biologist can calculate how many birds will get malaria when he allows infected mosquitoes to bite 100 healthy sparrows. यानी कितने लोगों को मलेरिया होगा if he allows the infected mosquito to bite 100 healthy sparrows. वो 100 में से कितनों को होगा? लेकिन प्रीवियस एक्सपेरिमेंट वो परफॉर्म कर चुका हुआ था. In the previous experiment, he noted that when he allowed to mosquitoes to bite 20 healthy sparrows, out of 20, 14 suffered from malaria. यानी कि दो एक्सपेरिमेंट हो रहे हैं एक्चुअली. एक साइंटिस्ट एक्सपेरिमेंट पहले कर चुका हुआ है and he has performed experiment on 20 sparrows and he allowed them to bite the malaria infected mosquitoes और 20 में से 14 को मलेरिया हो गया था. तो इस रेशो को फॉलो करते हुए the scientist is going to calculate how many sparrows will get malaria out of 100 healthy sparrows. यानी पहली रेशो तो ये थी ना कि 14 got malaria out of 20. अब सेकेंड रेशो हमने फाइंड आउट करनी है कि 100 में से कितनों को मलेरिया होगा. The x is unknown in the second ratio. Look how we arrange these two ratios with the sign of proportion. 14 out of 20 suffered from malaria. How many out of 100 will suffer from malaria? So this is how we used to solve our problems in early classes. 14 divided by 20 because ratio is equal to division symbol and proportion is equal to is equal symbol, equality symbol. So 14 divided by 20 is equal to x divided by 100. Now x is unknown to us, so we can find out the value by taking 100 on the other side. और even हम यूँ कह सकते हैं कि दोनों साइड को 100 से ट्रीट किया. एक साइड पे तो 100 क्रॉस हो गया, दोनों साइडों को 100 से मल्टीप्लाई किया. एक साइड पे तो 100 खत्म हो गया, तो दूसरी साइड पे 100 रह गया. So the answer is 70. I'm not a maths teacher, so I think I'm not good at teaching maths. So let's see. Although it's very easy, the answer came is 70. Out of 100, 70 sparrows will suffer from malaria, according to his first experiment. हालांकि बायो में बहुत ज़्यादा रेशियो अप्लाई नहीं हो सकती कि अगर पहले एक्सपेरिमेंट में 14 का हुआ तो ज़रूरी नहीं है कि दूसरे एक्सपेरिमेंट में 70 को ही हो. लेकिन ब्रॉडर पर्सपेक्ट में you can apply your results to a larger population. You can do that. जब वो कह रहे होते हैं कि out of 10 individuals, 2 are suffering from malaria, 3 are suffering from hepatitis, or I'm sorry for these जमाइयाँ वो काफ़ी देर से जागी होती हैं तो नींद आने शुरू हो जाती है. तो जब भी हम कह रहे होते हैं कि 10 में से 4 बीमार हो गए, 10 में से 2 बीमार हो गए, 10 में से 5 बीमार हो गए, तो this is actually ratio and proportion. But here I'm just talking about ratio. So these are the things which makes data analysis easy. Ratio and proportion is one of the examples. Otherwise, there are statistics. In statistics, there are so many formulas which can be applied to summarize the data. But these techniques are used to summarize data through the calculation of a mean value or any kind of formula. I mean, mean क्या है? Mean statistical technique है ना कि अगर mean average में हम क्या करते हैं? हम अगर पूरी क्लास के मार्क्स को देखें, लेट्स से बायो में मार्क्स आए किसी के 35, किसी के 30, किसी के 45 आउट ऑफ 65, किसी के 60, 62, 63, सारी क्लास की लड़कियों के मार्क्स को प्लस कर लिया और लेट्स से उस क्लास में 30 लड़कियां थी. तमाम मार्क्स को वी आर गोइंग टू ऐड एंड देन डिवाइड इट विद नंबर ऑफ स्टूडेंट्स व्हिच इज 30. So we get the data which is called as average, and that average is actually the average of the whole class. This is the average of this class. तो मीन वैल्यू क्या होती है? मिड की वैल्यू. जब आप पूरी क्लास के डाटा को एक लाइन में अरेंज कर लें, लेट्स से एक सबसे कम नंबर थे 22, फिर 23, लेट्स से फिर थे 28, फिर थे 34, फिर थे 40, 41, 42, 45, इस तरह से आपने अरेंज कर लिया डाटा को, and you find the middle value, and that middle value will be called as mean value. This is how, by using such simple formulas and techniques, you can summarize your data. This step is very important as it transforms our raw data into a meaningful information, which can be used to summarize and report results. I mean, by using these formulas and mathematical and statistical techniques, we can easily report them in our results, in journals, or we can publish them easily. Otherwise 200 पेशेंट्स का डाटा कौन पब्लिश करेगा? 5-6 पेजेस अलाउ करते हैं डाटा वाले, ओ जर्नल्स वाले, कोई 7 पेजेस अलाउ कर देता होगा इन अ जर्नल, बट 200 सफ़े अलाउ नहीं होते फॉर वन रिसर्च. You have to summarize and minimize your data. So you are going to use by different formulas and techniques, right? The next topic is mathematics as an integral part of scientific process. So ratio and proportion also seems maths to me, but these are simple statistical techniques. Mathematics is also a very important part of scientific process in every subject. So is the case with statistics. These techniques are going to be used by every science during research process, whether it's Park studies, math, even maths, um, physics, chemistry, psychology, any science, any field of science or arts, whatever the name you take it, you have to summarize the data by using statistical and mathematical techniques. So biological method also involves the use of applied mathematics to solve biological problems, because mathematics is an integral part. You have to summarize your data. So major biological problems in which the knowledge of mathematics is used, including gene finding, protein, protein structure, protein-protein interactions, okay? Bioinformatics में बताती हूँ, refers to the computational and statistical techniques for the analysis of biological data. Basically, at this age in which class you are reading, you cannot easily understand gene finding or protein-protein interactions. But I must tell you, this happens in the field of bioinformatics mostly. Bioinformatics is a branch of biology where actually you use all the computational and statistical techniques to find out protein structure three-dimensionally, or gene structure, or protein interactions. अच्छा, coming back to the basics again. अच्छा, यानी मैंने briefly बताया कि what is bioinformatics. Bioinformatics में भी degree हो रही है. I think so. I haven't told you in careers in biology. Okay, it's a kind of a degree which use computer का knowledge और statistical and mathematical techniques को use करके बायोमोलिक्यूल्स को ड्रॉ करता है आर्टिफिशियली. ये जो हम वीडियोस देखते हैं कि हाउ proteins are entering into the nucleus or leaving the nucleus, and how they are playing with the DNA, or sometimes you might have seen any video. That is actually just imagination. You cannot make a video at this level, at so much microscopic level. But you can make a video at certain level, but not to the level which I have seen. वो सब artificial होती हैं. You use computational techniques. So bioinformatics is a branch of biology which deals with all those computational and statistical techniques to find out the structure of biomolecules, interactions of biomolecules, उनकी bonding, उनके angle and all that. Look at this diagram even. This is protein-protein interaction. Let's imagine कि different proteins हैं. I have told you earlier, or if I haven't, proteins are made up of amino acids. प्रोटीन एक बहुत लॉन्ग चेन कंपाउंड है, लंबी सारी चेन है. उस चेन में आप सोचें, आप ये इमेजिन करें लंबी सारी माला है और उस माला में बहुत सारे बीड्स हैं. वो जो माला के जो बीड्स हैं वो अमीनो एसिड्स हैं. तो एक अमीनो एसिड, दूसरा अमीनो एसिड, तीसरा अमीनो एसिड, तो एक पूरी माला बना लेते हैं। बट उस माला के मुख्तलिफ एंगल्स होते हैं, ये गुंजलक बन गए, क्या कहना चाहिए, सबसे कॉम्प्लिकेटेड स्ट्रक्चर्स बनते गए। ये जो स्ट्रक्चर्स हैं, ये माला ही समझ लें, उसकी बीड्स डिफरेंट एंगल्स में ड्रॉ हुए, 3 डायमेंशनल स्ट्रक्चर बन के प्रोटीन बने। डिफरेंट प्रोटीन्स आर इंटरेक्टिंग विद ईच अदर। अब ये देखिए आर्टिफिशियल डायग्राम, हु इज मेकिंग दिस डायग्राम बाय यूजिंग डिफरेंट वेबसाइट्स। तो बायोइन्फॉर्मेटिक्स में डिफरेंट वेबसाइट्स हैं, डिफरेंट बैंक्स हैं, जीन बैंक्स हैं, जहाँ पर जो लोग भी सीक्वेंस फाइंड आउट करते हैं, जीन की सीक्वेंस है। उससे पहले मैंने आपको बताया था कि जीन इज अ स्मॉल सीक्वेंस ऑफ डीएनए। हैव आई टोल्ड यू समवेयर? जेनेटिक्स में बताया था ना? यस। तो जो स्मॉल सीक्वेंस ऑफ डीएनए है, ऐसे ही तो नहीं पता चल जाती ना, उसको किसी लैब में फाइंड आउट किया जाता है कि ये क्या है, फिर क्या है, आगे क्या है। लेट्स से कुछ सीक्वेंस है, फिर उसकी मैथमेटिकली बहुत सारी टेक्निक्स यूज होती हैं बायो में, जो कि आपके इस लेवल पर आई कैन नॉट प्रॉपरली एक्सप्लेन, बट यू कैन सी this diagram of a protein interactions, and look at this diagram as well. ये जो different, look at this diagram, ये प्रोटीन के स्ट्रक्चर्स हैं, सब यूनिट ए एंड सब यूनिट बी कंप्यूटर से बनाए गए हैं ना, राइबोन्यूक्लियेज एंजाइम है, व्हिच यू डोंट नो। तो यानी एक प्रोटीन के अमीनो एसिड के साथ डिफरेंट चेन्स हैं, तो एक चेन का ये स्ट्रक्चर बता रहे हैं, दूसरी चेन का ये स्ट्रक्चर बता रहे हैं। सो इज द केस ओवर हियर, डिफरेंट स्ट्रक्चर, विटामिन डी बाइंडिंग प्रोटीन, कोई प्रोटीन है जो विटामिन डी के साथ बाइंड करती है, उसका स्ट्रक्चर बताया हुआ है कि एक चेन ऐसे है तो दूसरी चेन ऐसे है। तो ये क्या है? ये भी किसी एंजाइम का प्रोटीन है, खैर प्रोटीन है कि जिसकी एक यूनिट इस तरह से होगी तो दूसरी यूनिट ऐसी होगी। So how can we draw these structures and how can we draw these interactions? ये interactions हैं of a protein, ठीक है? This is done by using different computational techniques, and it happens in bioinformatics, and that these techniques are also used by other scientists. Like this, these techniques are used in genetics. These techniques are used in enzymology, protein study, right? So this is also all, everything is deriving from mathematics. All these laws and principles to draw a diagram are being derived from mathematics. So mathematics is a important and essential part of scientific process. Without it, we cannot do a good research. Okay, so research is incomplete without mathematics and statistical tools and techniques. You have to somehow use them. जितना ज़्यादा आप उस लेवल पर आप समझ जाएंगे कि मेरी रिसर्च वर्क में ये वाली मैथमेटिकल टूल्स एंड टेक्निक्स यूज़ करूँ और ऐसे ऐसे मैं बताऊँगा, ऐसे ऐसे मैं एक्सप्रेस करूँगा, the more your scientific research will be reliable and good and productive. Okay, so Allah Hafiz from now on today. This is the end of second chapter, and I'm wondering what I'm going to teach next, because the syllabus for your class for the whole 8th class is only 4 chapters. First 4 chapters, chapter number 1, chapter number 2, 3 and 4. Although the chapter number 4 is quite big, but the whole year is left yet. So I'm astonished what I'm going to teach next, sir. Because according to the instructions given to me, they have asked me to teach only chapter 1, which was quite difficult to teach electronically, and you have no obstruction, you have no, I mean, kind of problems जो कि normal class में होते हैं कि कभी एक बच्चा बोल रहा है, कभी दूसरा बच्चा बोल रहा है। तो I think so वो तो बहुत ज़्यादा कम उन्होंने बताया था chapter number 1. Now I have to figure out what I'm going to teach, and I have to consult them as well. But there is one idea in my mind to teach how to make diagrams. I mean, I have to create 10 minutes video, 10, 10, 10, because I don't know if those videos will be uploaded easily. 15 मिनट की या 20 मिनट की वीडियो बना के उसमें बताऊंगी कि how to draw animal cell, how to draw a plant cell, how to draw a nucleus, how to draw a chloroplast. So maybe we can save time from the course of fourth chapter, और मे बी बीच में हम प्रोकैरियोटिक और यूकैरियोटिक का टॉपिक पढ़ लें, फाइव किंगडम क्लासिफिकेशन पढ़ लें. आई डोंट नो, आई हैव टू figure it out what next will be our course. So Allah Hafiz from now on. क्या हो रहा है? I'm saying pause, share, stop, share. Allah Hafiz.